Well, good evening, everyone, and welcome to the Royal Society. About a year ago, the Royal Society decided to start a series of international lectures where we would, by sort of rotation, invite scientists from different countries to present their work. And today, we're very happy to present Professor Wolfgang Baumeister from Germany. And the Society has always had a strong relationship with Germany, and as two of the leading scientific nations, especially in the EU, scientists in the UK and Germany have had very close links, and we at the Royal Society are in the process of persuading, or well, I think we have successfully persuaded our government at least, that it is in the interests of everybody for the UK to continue to maintain very strong links with the EU and, of course, with Germany in particular. And I'm delighted that Wolfgang Baumeister, one of the most foremost scientists in Germany, is with us today to give his lecture. Wolfgang received his PhD from Dusseldorf and then spent most of his career at the Max Planck Institute for Biochemistry in Martinsried, where he has been for the last several decades. Although I believe he did spend some time in the UK as a Heisenberg scholar in Cambridge. And he's known for a number of things, but the two areas that he is best known for is he's a pioneer in trying to understand the cellular pathways of protein quality control, and in particular, the structure, function, and regulation of the proteasome. And the other thing he's known for, which is going to be the topic of today's work, is that he's a world leader in the development of cryo-EM tomography. Now, many of you know or have heard of the cryo-EM revolution, including the improvement in resolution that makes possible atomic structures of individual molecules. But a second aspect to this, and one that is really becoming increasingly used and will become even more powerful in the future, is the ability to visualize molecules not as isolated molecules, but as they exist in the cellular context. And what that tells us about the organization of the cell and the role the molecules inside cells. And as I mentioned, Wolfgang has been a pioneer in this. Many of his proteges have gone on to use this technique in various faculty positions all over the world. And I'm very happy that he's going to be telling us about this, his work today. So.
Yeah, thank you very much, Venki. Thank you for the invitation. It's a pleasure and an honor to be here today. So I will, in the next 45 minutes, tell you about investigating the structure of molecules inside cells. That is the official title that was given by the Royal Society, or briefly, to do structural biology in situ. Now, why do we want to do that at all? I think it's fair to say that traditionally, structural biologists take an extreme reductionist approach. Cells are taken apart, the molecular components are fractionated, they are purified, and then they can be studied by any of the methods structural biology has at its disposal, whether it's X-ray crystallography, cryo-EM, or NMR. Now, this approach has been, of course, extremely successful if you look at the PDB. But it's also true that very rarely biological functions can be attributed to individual molecules. Biological functions arise from the interaction between the many individual molecular species inhabiting cells. So I think it is clear that many of these interactions which underlie cellular functions exist only transiently. Many are organized in the form of supramolecular complexes, but many of these supramolecular complexes are so deeply rooted in the cellular environment that it's very difficult, if not impossible, to isolate them without, in fact, affecting their structural integrity. So I think what structural biology in situ aims to achieve is to study macromolecular, supramolecular structures in their functional context. This means inside unperturbed cellular environments. And the method that enables doing that is cryo-electrontomography. I don't think with this audience there's any need to explain the principle of tomography. I mean, it's shown on the left-hand side. Now, I think it may be sufficient to say that cryo-electrontomography combines the power of high-resolution 3D imaging with the best structural preservation that is physically possible to achieve. The only preparation step is rapid freezing, such that the structures, the cells, become embedded in a thin layer of amorphous ice. Now, the idea to do something like electron tomography of unstained biological material is not new. There was a remarkable paper published in 1968 by Hart. He was at Lawrence Livermore Laboratory. The paper was entitled Electron Microscopy of Unstained Biological Material, the Polytropic Montage. And as you read the abstract, it's clear the polytropic montage is synonymous to tomography. And the final sentence in the abstract says, preliminary results indicate that one may thus study unstained, unshadowed biological material at high resolution. The paper had minimal impact, one should say, and the reason for that is it was the only application, was an application to freeze-dried cellular material. And as a matter of fact, there were no recognizable features in this structure. So it was largely ignored. But the vision was there, but for a vision to become reality, also the technology must be there. Now, very briefly, the problems in electron tomography are simply to obtain a detailed and an undistorted reconstruction. A tilt series should cover as wide an angular range as possible with increments as possible, meaning we want to maximize the number of projections. That, I mean, determines the quality of the tomogram. At the same time, this is difficult to reconcile with the need to minimize the cumulative electron dose to avoid radiation damage. So the solution to the problem is fairly simple. It's what we call dose fractionation. So instead of spending the entire allowable electron dose on a single image, which is then statistically relatively well defined, we distribute it over as many projections as possible, which are noisy, and we can do it only to the extent that they can still be aligned with respect to each other, with respect to a common frame of coordinates. Now, so we began to work on the automation of electron tomography in the late 80s. At that time, this was not just a matter of software, also hardware had to be built in order to enable doing it. And it was basically in 91, 92 that we could show that the cumulative electron dose in collecting tomographic 3D data sets can be kept small enough for the investigation of ice-embedded specimens. This was the first cryotomogram we published, something as trivial as lipid vesicles embedded in ice. But it showed that in principle this is possible. We went a little bit further. We encapsulated in the lipid vesicles actin, let it polymerize, so it stretched out the vesicles. And we did a reconstruction. I'm showing it only to make the point that, that because of the dose limitation, tomograms are noisy. And one way of dealing with that is to develop denoising methods, basically meant to improve or to maintain the signal, but to get rid of the noise. Now, there's a more powerful way of dealing with the noise. Whenever a tomogram contains repetitive features, we can do what is called subtomogram averaging. And this is an early example, maybe the first example of doing subtomogram averaging. It was a reconstruction of a herpes simplex virus particle. And since it contained an icosahedral capsid, one could easily then average the icosahedral capsid and put it back precisely into the context. Now that is now a common approach in cryo-electron tomography. Now we began at about that time also to grow cells, all kinds of cell types directly on EM grids, and basically all I'm going to show in my talk today is about cells grown directly on EM grids. The problem is, these cells were dictyostelium cells. The problem with growing cells on a grid is that only the periphery of the cell is transparent to the electron beam. I mean, anything that is thicker than half a micrometer or a micrometer remains in the dark. It's not transparent to the electron beam. So it is clear that in order to achieve a decent resolution, one must apply some sort of thinning. There are various methods. What we want to avoid is subcellular fractionation. You can, of course, many organelles isolate easily and do tomography on them, but that is not exactly what we would like to achieve. The same is true for laser microdissection. Cryosectioning can be applied in principle. I think Jacques Dubochet's lab has worked on that for a while. It's very tedious. It's very difficult to convince a graduate student to do it. But the real problem is that that amorphous ice is quite compressible. So, I mean, if you physically section with a knife ice, you get compressions amounting to something like 30 or 40%. And that means subtomogram averaging is not really applicable anymore. So we put all our resources in terms of personnel, but also in terms of money on focused ion beam micromachining. Okay, so I mean, yeah, that is a brief definition of what we call structural biology in situ. It should apply only to a scenario in which the cellular environment is preserved in its entirety. All organelles, all supramolecular assemblies are kept at their native place. And the method that allows doing that is focused ion beam milling. So you see here, again, dictyostelium cells grown on a grid. So we have them in a dual beam instrument, which has to be modified, of course, in order to be cryo-compatible. So we have an SEM beam, where we can observe what is going to happen. And we have a focused ion beam, a gallium beam to ablate material. And so we can use that method to cut, we can apply various different geometries. We can cut a kind of a ramp or a wedge. But I think the approach we are using mostly is the one shown here, that we ablate material from above and below. So leaving behind essentially a thin lamella that is supported only by the surrounding ice. So we typically cut lamella which have a thickness between 200, 300, 400 nanometers, depending on what we want to accommodate in the structure. I think you have heard, and Wenke mentioned it, in recent years we have seen enormous advances in EM technology. I think one should not forget that an important aspect is the robotics that is in today's electron microscopes that makes automation quite a bit easier. It's just a matter of software nowadays. So very large data sets, I mean, microscopes can be operated more or less autonomously. We can obtain very large data sets of fairly consistent quality. Now, I think the direct detectors, I mean, were game-changing in the sense they are much better detective quantum efficiency. You can collect data in a movie mode, as many of you will know. We can correct for motions. And last but not least, I would like to mention the faceplate that was developed by Radostin Danev in the lab. A little device that is put in the back focal plane of the objective lens. And it allows, I mean, it acts similarly to the Cernica faceplate in light microscopy. Now, as you all know, I mean, biological material embedded in ice, if you take an image in focus, there's hardly any contrast. So what people do is they generate phase contrast by defocusing, but that results in a weird contrast transfer function here or in a ghostly image. And whereas the Volta faceplate now allows to collect data or to generate phase contrast, sorry, in focus. I think I show a first example of some applications combining all these technologies on a project that is still very much in the beginning. We are interested in the structure of the mouse retina rod outer segment. So this part here, and we are interested in particular in the supramolecular organization of the disc membranes. Now, I'm showing that because it's a particularly striking example comparing on one hand, what we were able to do about 10 years ago. You see that the isolated rod outer segment is just a little bit too thick, so we don't see a lot of detail there. And this is now a rod outer segment after focused ion beam milling and taken now with direct detectors and the Volta faceplate and so on and so forth. I think you will appreciate already, I mean, looking at the raw data, that there is an enormous amount of information, but mining this information is very challenging, and we are only making very slow progress here. So there are certain things which are straightforward, like the structure of these of the periphery of these disc membranes, which have an incredible curvature. There's a protein called peripherin that enforces that curvature, maintains that curvature. We see a lot of information if you look very carefully regarding the linkers, which determine the supramolecular organization of these structures here, which is remarkably well ordered. We are making progress with this one here because we have now isolated peripherin, now doing a single particle structure which we can dock into the tomogram. But the main challenge is to look for the supramolecular organization of rhodopsin in the membrane, and we are at the very beginning here to interpret the modulations in density in the membrane plane. But I'm pretty confident we have now good structures of rhodopsin dimers and so on that we can, in the end, interpret these tomograms completely. Now, a last slide on methodology. All I have described so far can easily be applied to cells grown on a grid or deposited on a grid, but it's a different matter if we want to look at something that is in a tissue or in a multicellular organism, such as C. elegans in this case. So here we have to cut basically vertically with the focused ion beam into the sample, and then we have to lift out basically this block of ice, which is typically between 2 to 5 micrometers thick initially. With a cryo gripper, we transfer it as shown in this little movie here. We have to transfer it to a second holder, and then we can, in the focused ion beam, mill it further to the desired thickness. So what you see here is a tomogram of a cell from the gut of C. elegans, and you see in these magnifications here, these are coated vesicles. These are ribosomes sitting on the endoplasmic reticulum. If we average them, we can go to a resolution of 10 angstroms or so, or microtubule. So this method, which is still not routine, I have to confess, but it enables us essentially to target any volume, even in a tissue or in a multicellular organism. Now, an application that has been published is something a HeLa cell grown here on a grid. You see here, basically that is the stained nucleus. So we cut a slice that is located at the periphery of the nucleus. And so we can now take a tomogram, and as we go from the top to the bottom, here we are inside the heterochromatin, and as you look very carefully, you will see individual nucleosomes with remarkable clarity. It's no problem to average them to a decent resolution. Then as we move a little bit further inside, we come to the fine meshwork of the nuclear lamina. This is an individual nuclear pore complex on the nuclear side, the same one now on the on the cytoplasmic side of the nuclear envelope, and here we are on the outer surface of the nuclear envelope where you see rows of ribosomes or polysomes. Now we can analyze them a little bit more deeply, so we can apply, after some denoising, segmentation techniques for microtubules, for actin, for the nuclear meshwork of the nuclear lamina. I don't want to go into too much detail, but maybe I draw your attention to this structure here. It's a nuclear pore complex. It's not the highest resolution that has been achieved, but it's remarkable in that it is a structure that was derived from averaging over only 4 individual nuclear pore complexes. So you see the difference here between the nuclear pore complex without the faceplate and here with the faceplate. And I think I think it will be quite rewarding to push it further such that we can, in the end, get a structure of that kind. Even if it's not the highest resolution, but from an individual nuclear pore complex. The reason is they vary tremendously. You see here at the bottom, they can change in diameter easily by something like 13%. So it does not make sense to really average over many individual nuclear pore complexes. So that is basically a summary here. So again, you see in the background the heterochromatin, then you see the fine meshwork in reddish orange color of the nuclear lamina, and on the surface, on the outer surface, you see the polysome sitting. Now, one of our favorite organisms is, for a couple of reasons, Chlamydomonas. And what you see here is basically now a lamella accommodating, a slice across an entire Chlamydomonas cell. What we can do now is we can zoom on any part of this cell here. So here we have the nuclear envelope. You see here, somehow it's a bit difficult for me to see from here, the nuclear pore complex. Here we have a Golgi apparatus. Here we have a piece of endoplasmic reticulum studded with ribosomes. Now we can go into such a tomogram and extract the features of interest. For instance, we have given these tomograms to John Briggs, who had worked on the COP1-coated vesicles and had done a reconstruction of reconstituted ones. And he wanted to see whether the in situ structure, the native structure, recapitulates the reconstituted one, which it does. But in addition, of course, in situ, these vesicles are filled with cargo, and also the topological relationship to the Golgi is preserved, and this means we can follow the biogenesis of these vesicles. We have given the data also to Martin Beck, and Martin has generated averages of the nuclear pore complex from this alga, which has some interesting features. It's quite different from that of, let's say, a yeast cell, even though the building block, the so-called Y complex, which is a subcomplex formed by something like 10 different polypeptide chains, is the same. The oligomeric state and also the topology is different. And as a result, the pore in these nuclear pore complexes is much wider open. It's something like 65 nanometers in diameter versus 45. So I have to show a ribosome. This is a ribosome extracted from the cytoplasm of Chlamydomonas, and this is the average. I think something like 800 or 900 particles, but one cannot compare it with numbers from single particle analysis because each of them is a 3D structure here. I think without, pushing the envelope really, this is a respectable resolution of something like 8.5 angstroms, and we can do that for ribosomes in all organelles we like. Now, well, finally, this is from work of Friedrich Förster in the lab. This is the structure of ER-associated ribosomes on the Sec61 protein-conducting channel, and the resolution is good enough, at least, to see very clearly the helices in the protein-conducting channel. But our interest is not ribosomes, but the opponent of the ribosome, destroying proteins. As Wenke mentioned, we have been working for many years on the proteasome system, which is, the executive end of the ubiquitin proteasome pathway of protein degradation, degrading proteins that are no longer needed or misfolded, and so on and so forth. So it's a multi-subunit complex of 34 different subunits. And this slide is just a reminder that 5-6 years ago, the basic architecture was solved by interpreting a 7-8 angstrom density map of the complex in the light of data that came from cross-linking and mass spectrometry. Now that is no longer needed. We can go to quite a bit higher resolution now. This is the structure of the human proteasome at 3.9 angstroms here. Now this is obviously not in situ, but I need to introduce the structure in order to explain what we see in situ. So the red part is the 20S proteasome, and the structure had been determined by various groups in the meantime by X-ray crystallography. That is a very sort of robust entity, but it has to associate with a regulatory particle here, which essentially prepares substrate for degradation, which means it recognizes ubiquitinated proteins, it deubiquitinates them, which is a prerequisite for unfolding. It unfolds them, and that is all done by the blue part here, which is a AAA ATPase, which is at the core of the regulatory particle, and it also assists in translocating substrates into the core complex. Now, what is important for what I'm going to talk about in the next part of my talk is that the 26S proteasome is a highly dynamic structure. It undergoes very large-scale conformational changes during its functional cycle. There's now pretty good coverage of the different conformational states it undergoes during its functional cycle. I mean, we are now at something like 6 distinct conformational changes. I mean, this paper has just been accepted today. I think I will be very brief. I mean, essentially we have 3 basic states. S1 is the ground state. That is the state in which it's not dealing, not transacting on substrates. It's sitting there and waiting for substrate. Then there's S2, which we call the primed state, or it's a commitment state. It means the substrate is, or the proteasome is engaged with the substrate to an extent that the whole process of degradation becomes irreversible. But then it continues to the substrate processing states, and the large-scale conformational changes, these rotations here, but also shifts, serve basically to bring the substrate, which is initially at the receptors, down to the mouth of the AAA ATPase for unfolding. But also, there has to be an alignment between the unfolding channel in the AAA ATPase complex and the gate that controls access to the core particle. So that is basically the shift we see here. So the important thing for understanding what I'm going to tell you now is that the conformational changes are of such a magnitude that we can relatively easily, even at medium resolution, let's say 15, 20 angstrom, but we can go quite a bit further than that now, we can infer from the conformational state what the activity status of the proteasome is. So where do they occur in the cell? Now in Chlamydomonas, but also in yeast, very roughly half of all proteasomes are in the nucleus, and the other half is in the cytoplasm. Now in the cytoplasm, they appear to be basically everywhere in the cytoplasm. There is no preferred orientation or positioning. However, invariably they associate with the endoplasmic reticulum, and the endoplasmic reticulum occupies roughly 30–40% of the volume of the cell. This means that is why they are found essentially everywhere. But they are clustered, as you will see in a minute. In the nucleus, the vast majority of proteasomes is in vicinity of the nuclear envelope and at very distinct sites. Now, so these are the raw data, and again, I think I cannot see it well from here, but you might be able to see it even on the raw data. You can recognize the characteristic features of the 26S proteasome, shown with these yellow arrowheads. This is a region near a nuclear pore complex. So we can do subtomogram averaging, and in this case, it's a sort of an intermediate resolution because we had a relatively limited number of particles here. But you can not only, generate a global subtomogram average, you can also do a lot of classification, as I will illustrate further on. And then we can distinguish whether they have one or two regulatory particles associated. We can infer, as I said, from the conformation whether they are engaged with substrate or not. Now, the large majority in the nucleus is in the vicinity of the nuclear pore complex. And we have two distinct populations of 26S proteasomes there. In red, that is a population that is relatively close to the membrane. It's tethered to the membrane, as you will see. And in yellow, that is another population which is further away from the membrane, but it's closer to the eightfold central axis of the nuclear pore complex. So we can map back them to individual nuclear pore complexes, the position of these two classes of proteasomes. Those that are further away, they are directly tethered to the nuclear basket. So you see the nuclear basket has, of course, eightfold symmetry, and we never have full occupancy, but we might have 6 or 7 of the positions decorated with proteasomes. And as I said, we can now distinguish between those which have 2 regulatory particles, 1 regulatory particle. We can say which one is in the ground state and which one is in the substrate processing state. And we can also say whether it's a free one or it's tethered either to the basket or to the membrane of the membrane envelope. And we can map them back. So we show here again a raw tomogram. And you see in this region here, we have a mitochondrion. Here we have the nuclear envelope. Here we have nuclear pore complexes. Now in blue, we show all proteasomes. Now, as we change color, we have single capped versus double capped. Then in the next one, we will have those in green, which are in the ground state. And this reddish color, those which are in the substrate processing state. The free ones are in light blue, and the basket tethered ones are in yellow. So we get a lot of information from that. And basically, this is summarized here. So we have these 2 classes we expect, and we are trying to identify currently the tether so that we can knock the tether out. We have good candidates for that. So we assume that this one is more involved in the surveillance of import and export. And this one here probably has a role in maintaining the different protein composition of the outer and inner membrane of the nuclear envelope, because this is the only place where we have continuity between these 2 membranes. Now, coming to the ER-associated proteasomes, I think many of you have heard about the unfolded protein response or ER-associated degradation, ERAD system, which is well studied and characterized biochemically. So basically, the bottom line is we have the endoplasmic reticulum, ribosomes sitting on the ER produce protein, which is translocated into the ER with the assistance of molecular chaperones, and they are ideally perfectly folded. If they are perfectly folded, maybe also modified by glycosylation, then they become exported from the ER. However, if things go wrong and we have aggregation, then and we have the unfolded protein response under stress conditions, we have retrotranslocation from the ER, and then they are degraded outside the ER by the proteasome. Now, there are many more players involved because, I mean, also the ubiquitin system has to become involved. Now, as we look at the endoplasmic reticulum here, there's a region of the endoplasmic reticulum. What we see is that we don't have, I mean, single proteasomes sitting on the surface of the endoplasmic reticulum, but what we have is we have clusters of proteasomes sitting there, and these clusters segregate very well from the regions of the AR, which are occupied by ribosomes. Now, well, that might be a little bit hard to see, but I think you can again recognize, even in the raw data proteasomes, you can recognize the AAA ATPase CDC48, which is implicated and believed to be involved in the translocation in what way ever. I think that is a bit controversial. And we have, of course, ribosomes free and ER bound. I mean, all these clusters have similar features. They have similar size. Typically, the diameter of these proteasome clusters is something like 200 nanometers, so they accommodate between 20 and 30 individual proteasomes. And so we can do quite a bit of more detailed analysis here, a molecule by molecule analysis. So if we have a template of the structure that is under scrutiny, then we can use it to perform a spatial and angular search, and this means it gives us the positions of the molecule in the cell. Then we can extract the molecules, and we can do what I described already, subtomogram averaging, and we can map them back. But if we have different species or heterogeneity of any sort, basically, then we can classify them into classes according to their similarity, and we can generate subtomogram averages from the different classes. Now you see that here applied to the clusters of the endoplasmic reticulum here. It's a reference-free alignment, so as a reference we use here basically only a blob which converges to the structure of CDC48. Now that structure is not very well resolved, and the reason is that CDC48 is in these clusters a relatively low abundance protein, so the structure of the proteasome and the ribosome is much better defined here. The interesting thing is that the method is remarkably quantitative. So we have compared here some mass spectrometry of Chlamydomonas looking for proteasome CDC48 for ribosomes. So when we simply count the ribosomes after template matching here in the tomograms, then we get basically the same concentration as we get from mass spec. The same is true for much less abundant CDC48. Proteasomes, there's a difference, but the difference is due to the fact that in this experiment we counted only proteasomes in the cytoplasm. And as I told you before, roughly half of the proteasomes are in the nucleus. So the method is quite quantitative, which gives us confidence that we don't miss too many of those particles. Now a little bit about the clusters here. This is another 2 clusters, and if you do statistics, you see it's a very clean separation. So where the proteasomes are, the ribosomes are excluded. And CDC48, that is, I mean, down here might be a little bit difficult to see, is basically at the interface. But it's sort of spread out. Now, again, we can do the analysis a little bit further, not only assembly states, we can also look at the functional states. It looks at a glance here that with outside clusters or inside clusters, the number or the proportion of those in the substrate processing state is the same. However, if you take a little bit a closer look and look at the proteasomes which are in immediate vicinity of the ER membrane here, then the fraction that is in a substrate processing state is much higher. So it's roughly 50%. And a little bit further away, it's still relatively high. And then, I mean, in the remainder, it's about the same level as outside the clusters. Now, all those which are directly adjacent to the membrane within 20 nanometers or so, they have some density, not very well defined, but they appear to be tethered to the envelope, to the nuclear envelope. To the endoplasmic reticulum. So it's more density than you would expect for an unfolded polypeptide chain. So there must be other factors being associated with it. But it seems that, and that had been hypothesized before, that the 26S itself can or plays a major role in extracting polypeptides from the endoplasmic reticulum. I think summarizing this part here, yes, we have clusters. The clusters are, we have at the periphery CDC48, but I think the important thing is that the ERAD system, the protein degradation pathway, is spatially quite nicely separated from the secretory pathway, which is always on the opposite side of the endoplasmic reticulum. I think in the last part of my talk, I will tell you about a project which is a collaborative project between 3 departments from our institute. Ulrich Hartel's group. Ulrich is interested in how do aggregates affect the proteostasis network? How can we boost the cellular defense against aggregation? My colleague Matthias Mann, who is a proteomics expert, is interested what is the composition of the aggregates, how does the cellular proteome change in response to toxic aggregation, and our interest is what is the in situ structure of the aggregates and how do they interact with the cellular environment. So that's a project that is funded by an ERC synergy grant. Now, I will begin with aggregates of Huntingtin. I think you all know about Huntingtin, and we have, a few weeks ago, or for a month ago, we have determined the structure of the full-length Huntingtin, which was by single particle analysis. Now, this has taken us 5 or 6 years, I must say, because the molecule is extremely flexible, and I think the only solution was to find an interacting protein here, HUB40, which I mean like a wedge inserts in between the domains, and that allowed then to go from 8 or 10 angstrom finally to 3.5. Now, Huntingtin is the normal Huntingtin, is involved in multiple cellular functions, including vesicle transport and so on and so forth. Now, I think what makes it a pathogen, if you like, is if you have, an excessive polyglutamine repeat in the exon 1. So in this region here, that would be a short one. So people who carry such a short repeat are protected from the disease. If that becomes longer than 36 or so polyQ repeats, it results in the formation of an unstable protein which tends to aggregate. Now, I think this is the beginning of a little movie which I will show only for a few seconds. Basically, what we have done is we have expressed a model Huntingtin, not the full length as a matter of fact, but with relatively long polyglutamine repeats, 60 and one with 90, and you see here a light micrograph of an aggregate. In green you see the Huntingtin inclusion here. I'm showing that to make the point that what you have seen is, maybe repeat that briefly, that the dynamics of the ER in the vicinity of the inclusion is sort of arrested. You see, there's a lot of dynamics in the part of the ER that is relatively far away in this region here, whereas surrounding the inclusion body, basically there's no motion. So the challenge is now, of course, to hit such an aggregate in tomography, and that requires a relatively complex workflow, which is not as well integrated yet as it should be eventually. So first step is always vitrification. The second step is correlative fluorescence microscopy. We use that to identify the feature of interest, to determine its coordinates such that we can transfer the coordinates to the cryo-focused ion beam and cut a lamella that includes the aggregate. Now, now we zoom into one such aggregate. In this case, is an aggregate expressed in a HeLa cell, and you see the very fine fibrils. They are sort of radially arranged here. Again, I think this would not have been possible without the new detectors, without the faceplate. The fibrils are only something like 3 nanometers in diameter, so they are very fine. But quality of the tomograms is good enough to allow for automated segmentation. Now, we were a bit concerned that the presence of GFP might alter the structure, and it does, but it does in a relatively modest way. So, for instance, the length of the filaments is the same in the absence and presence of GFP. However, the density of packing, and that is shown in this histogram here, that varies. I mean, in the presence of GFP, it's less densely packed than in the absence of GFP. You see that on the next one here, that is a Huntingtin fibril decorated, as a matter of fact, and you see the particles with GFP. On the right-hand side, you see the untacked Huntingtin fibril. So the density is different. In terms of stiffness, it's the same. Now, this is now a Huntingtin aggregate in a primary mouse neuron here. So again, this is the raw data. That is the segmentation. The same architecture, essentially, of the aggregate of the fibrils. You see the only, let's say, inhabitants of the aggregate are tiny vesicles, and we can show that they are derived from the endoplasmic reticulum. The endoplasmic reticulum, as I told you, which is not is kind of frozen in the surroundings, is disrupted at the periphery of these aggregates here. So in red, you see the remnants of the endoplasmic reticulum. And as a consequence, the ribosomes sitting on the ER are liberated. These are the green particles. The interesting thing is they remain completely excluded from the volume of the aggregate here. So the aggregate might be a kind of hydrogel of some kind, not letting, in this case, any macromolecular species intrude. Now, I think the mechanism of disrupting the endoplasmic reticulum is poorly understood at the moment. The fibrils seem to poke the membrane, or what we observe is, and this is, you see it easier here in this magnified view, whenever they make contact to the membrane of the endoplasmic reticulum, they dramatically change the curvature of the membrane, and that obviously leads to some sort of disruption. There are extreme cases, might be a little bit difficult. Here's a lysosome and here's a fibril, and it sort of pulls out, I mean, a large part of the membrane. We are trying to recapitulate these things currently in vitro to come to a more detailed understanding. A second system I want to describe briefly, and that brings us back then to proteasomes, is aggregates formed in the context of these two diseases, FTLD or ALS. The cause or the of the disease is repetitive hexanucleotide expansion in the gene. Now that seems to result in, to some extent, in the loss of function of that gene, but I think the thing we are interested in, it results in the production of D-peptide repeats here. And most inclusions in patients from ALS contain glycine-alanine repeats. So we have used a model system that recapitulates these features, and indeed it forms aggregates which are reminiscent of the disease aggregates. Now, again, that is the workflow, but shown now in a different way. So we grow the neurons here on the grid. We select a cell that, according to the GFP signal, contains the aggregate. Then we can very precisely target that in the focused ion beam, and we have and can control that here in the lamella. Now, this is now a tomogram through a polyglycine-alanine aggregate in a primary neuron. At a glance, it might seem similar in a way to the Huntingtin, but in detail, it's very different. The one difference is that you see many, already with the naked eye, that there are many particles somehow inhabiting these aggregate here. The second difference is these are not fibrils, these are ribbons. They are very thin polymorphous ribbons, often bifurcated. So that is one difference. Then, as I told you, I mean, there are many macromolecular complexes inside, and we can play the games which I described before in terms of the image analysis. So we analyzed it without using templates, as a matter of fact, for just picking particles and then going in through classification. What is in common with the Huntingtin is that ribosomes can never enter. They are always at the periphery here. Trick, which is the eukaryotic chaperonin working downstream of the ribosome, forms clusters in the vicinity of the ribosome, but the aggregate itself is densely populated with 26S proteasomes. It's a roughly 40-fold increase in the local concentration of proteasomes inside the aggregate. That essentially depletes the remainder of the cell of a key component of its protein quality control system. Now, since we have so many particles here, we can push the classification and the averaging quite a bit further. So this is a kind of an intermediate result, but you see these are in green are the ribosomes which are in the ground state, and here resolution is almost 10 angstroms. Then we have the substrate processing ones in blue, and that is inferred only from the conformational state. Now, invariably, those which are in the substrate processing state have, as shown here in purple, some additional sort of irregular sort of mass, which we tentatively identified as substrate. Now we can prove that and show that directly. We map now back the green ones and the blue ones, and so you will see the blue ones are always in direct contact with the aggregate material, as you see in this little gallery here. So they obviously try to degrade the aggregate material, but it's known that these glycine-alanine repeats, it's known from the defense mechanism of some viruses, they stall the proteasome. Obviously, these glycine-alanine repeats cannot be unfolded, so the system gets completely stalled. So we are pushing resolution here quite a bit further. Shanggu in the lab, who is working on that, has now collected a data set going at slightly higher magnification. So I think it's going to something like 7 angstroms now, but what we already see here is we can identify the subunit of the regulatory particle which is in contact with the substrate here, and that is RPN1, which is, I mean, a major hub also for shuttling ubiquitin receptors. So this makes sort of sense, but we want to push it quite a bit further. Now, to summarize and to come to the end here, this is a picture taken from a News and Views written by Wade Harper recently. He compares the two results. So we have the Huntingtin, which, I mean, obviously one toxic mechanism, not necessarily the only one, the oligomers could play a role as well, is the destruction of the endoplasmic reticulum, whereas in the case of the glycine-alanine repeat protein, the whole thing acts like a sponge, taking up all the proteasomes and compromising the protein quality control system of the cell. So that is our interpretation. Now that brings me to the end. I think the work on nuclear and endoplasmic reticulum is the work of Sarada Albert and Ben Engel in the lab. The work on the Huntingtin aggregates is the work of Felix Bäuerlein and Ruben. Friedrich Förster contributed a lot to the image processing, subtomogram averaging strategies. I've not talked about Foucault's work. Shanggu is the person who has done the glycine-alanine repeat story. Julia, I showed her data now at EMBL. He showed her data on the nuclear periphery. Antonio plays a role in developing segmentation tools. Radostein developed the faceplate. Jürgen Plitzko is in charge of many of the technology developments. Miroslava is the expert on focused ion beam milling, and Elizabeth Villa, now at UCSD, was quite a bit involved in developing the focused ion beam, and I mentioned collaborations here with Martin Beck and John Briggs. And I thank you for your attention.
Thank you very much. So, thank you for that really brilliant lecture. It's a real tour de force that combined, showed how you can combine methods development and new technological development to attack problems that couldn't be attacked before. And since we are here at the Royal Society, it does remind me of how many advances in biology have been made simply by being able to see things that you couldn't see before, going all the way back to Leeuwenhoek and Robert Hooke, which showed us that microbes and cells exist. And Wolfgang's lecture sort of shows that we're on the threshold of something similar, of being able to see structures in cells at a level of detail that we could never see before, thanks to technological developments, many of which are driven by Wolfgang himself. So thank you very much for that lecture. And perhaps you can take a few questions. So if there are questions.
Would it be possible, rather than using neuronal cell lines, would it be possible at some point to dissect a mouse brain and zoom in what you want to see there and do tomography?
Yeah. Well, I think I think the problem would start probably with vitrification. Okay, I mean, tell me how to vitrify a mouse brain. I think already, I mean, when you go to C. elegans, you have to move from plunge freezing to high pressure freezing, and high pressure freezing is of course already a bit more difficult to combine with focused ion beam technology. Yes, we had the problem when we wanted to publish the Huntington aggregate in Science. One reviewer said, well, I don't believe in constructs that are supposed to mimic the disease. So I believe it only if you take the brain of a patient who died of Huntington and offered brains from a collection they have at Harvard, which had been stored for decades in formaldehyde. Okay, so I think this, yeah, we defended it and we got it through then in Cell, and then Science decided to make it the editor's choice. Okay, yeah, I think that is, I think tissue, if you can, I mean, slices, if you prepare slices by some other means of a mouse brain, that would be thin enough and well enough preserved for high pressure freezing. I think then you could try doing that, okay? But I said that the lift out procedure is working. It's working in principle, but only few so far managed to make it work. We have not succeeded at all to convince manufacturers to provide instruments of that kind. So it's all home built so far. But I think in the hands of experienced people, it works quite well. So I think it's a remote possibility, but it would be possible. So far, we do essentially everything that is where we want to achieve some throughput. We do with cells grown on a grid, and neurons can be grown very easily on a grid. And I think it's much of the molecular biology that has been done on these disease models has been done on cell systems of that type.
Hi, very nice, thank you. I have a question, more general question. So you showed some really beautiful data, and which could be a gold mine for anybody wanting to study many different biological processes. On the other hand, it's the result of very hard work from a few people. So I wonder what your vision is regarding sharing the data? Should it be shared, and what's the best way to approach that?
Yeah, it's still a relatively complex workflow. The workflow could be simplified greatly. I think there's no reason that, let's say, the cryofluorescence microscopy and then the focused ion beam is in two separate instruments. This could be integrated very easily into one instrument, and the plans have been around since quite some time. So I think that would simplify things. Also, there's absolutely no need that the equipment that is needed for doing that costs in the range of millions. Okay, I think it's very clear that a bio-focused ion beam, we don't need these giant chambers and all that. I mean, could be built for something like 300,000-400,000. So that would make it accessible. That is one thing. On the other hand, I think there's also the option, as we have, indicated here. I think if you take over certain cell types, high-quality tomograms, they can easily be distributed in the community. Okay, so as we have done with some of our data, but that could be done in a broader way, so that basically people could sort of scan them for their PET molecules, so that it's only basically the image analysis that is done then by other people interested in a given system. So I think there are many possibilities. I think one should select probably 4 or 5 or more cell systems and generate, high quality data to be made available. That is would be my concept.
Do you think you could identify smaller objects in these full tomograms than proteasomes and ribosomes with further improvements in
CDC 48 is 400,000. Nucleosomes work quite well. It's 200,000. So it's quite a bit smaller. But of course, much below 200 kilodalton is currently dangerous, but the quality will be improved. I think once we go regularly, and I don't see any reason why we should not, to something like 7 angstroms, so that we clearly see, as the case in some cases, the helices, then I think you can go quite small. Okay, so I think probably it will become critical below 50 or so. At least during my lifetime.
In your nucleosomes in situ, can you see the connecting DNA?
No, that is a good. That is of course. So it's easy to extract them and to get decent structure of the nucleosome as such. But the problem is the connectivity, and that requires better image segmentation methods. There are 2 postdocs in the lab working on that, basically using model systems similar to what we have done some time ago with polysomes, okay, in vitro. So to learn basically, to improve the segmentation. So establishing the connectivity is currently it's not trivial or not safe. But this, of course, would be required to address higher order chromatin structure and to settle some of the disputes in the field.
So perhaps I could ask one last question. Where do you see the technique going in the next few years? There are obviously advances in detectors, the new generation of detectors,
I think
the other technologies. So what do you see as
I think it will not be a single development. I think it has to be, I think one thing where we are currently losing resolution is the precision of the alignment of the tilt series, especially with lamella, without fiducial markers. And so I think that is something one has to work on. I think one would like to have some sort, maybe ribosomes are good as fiducial markers. That is certainly one consideration. So then I think, yes, there's room for improvement on the detector side. There's room for improvement in the use of the faceplate, I think, and of course it would be relatively easy, or is relatively easy, to go to high resolution by going to even thinner lamella. Okay, I mean, the thicker, the more difficult it becomes to go to to a decent resolution. But there are certain, it's already relatively delicate to handle lamella of, let's say, 100 nanometers, but and it's always a compromise between, I mean, maintaining a certain volume and a thickness that allows to go to high resolution. But there are many issues you can address easily with if you go down to something like 70 or 80 nanometers thickness. Okay, so, but so it will be a combination of improvements of all aspects of the technology that are involved in order to bring it. I think it also, of course, the abundance of the molecules is an issue. It's easier with ribosomes or with proteasomes, which are relatively abundant, than with. But we have we have done decent structural studies with molecules as rare as tripeptidyl peptidase, for instance, where in one tomogram you have one copy. Okay, so then it becomes hard when you have to go to 70 tomograms or 100 tomograms in order to get a decent structure. Still, it's doable. Yep.
Very good. Well, thank you again very much. Okay.
I think I've